Hi, everyone. Thanks again for coming to today's session of Beyond the Scope. Before we get started with that, I just want to point out our upcoming talks that we switched to a schedule doing every other week for the rest of the semester. And we'll have 3 more talks coming up this semester. So in 2 weeks, we'll have the CEMAS Director and Professor of Material Science, David McComb, will be giving a talk about monochromated STEM, about some different functional materials and understanding how EELS works itself. Then we'll actually be going to Carly Goodwin, who will be talking about the Helioscan Micro CT, which ends up using quite a bit of the Avizo software that'll be discussed today. And then we'll finish up the semester going to Cryo-EM with Yoshi talking about how to do some data processing using CryoSpark software for single particle analysis. Today, we have Ali from Thermo Scientific. He'll give a talk on the Avizo software that we also have available here at CEMAS for use. I know Ali's been out here, he was out here last year. I think he gave also another seminar recently for just CEMAS affiliates. So it's been great that he's been out here before, knows what's going on, and I'm excited to hear a little bit more about the Avizo software and kind of see the basic capabilities that we can discuss with everyone. For the seminar itself, it should be around 30-40 minutes long. So in this time window, we'll have plenty of time for discussion and questions at the end. If you have any of those questions, feel free to put them in the Q&A throughout, and we'll answer them at the end, and we'll get to those as well. If you have any problems, go ahead, just put any questions in chat as well, and we can help out. Thanks. Okay, Shoj, I'll hand it over to you.
All right, thank you, Daniel, and thank you everybody for attending. I'm gonna go ahead and share my screen now. So what I'm gonna talk about today is essentially Avizo software for the analysis of both microscopic and tomographic images. This is a quick overview of all the topics that we're going to talk about. We're going to talk about the software that we have for image analysis. Then we're going to go briefly over the overall process of image processing or image analysis for scientific applications. From reading the data to quantifying the data, we're going to cover every single step. Then after that, we're going to talk briefly about the different things that you can do with your images. We're going to talk about how to work with porous materials, how to characterize them. We're going to talk about simulation with images, fiber analysis, performing digital volume correlation, and then essentially tracking mechanical deformation with just images. That's also, we're going to very briefly talk about that. And since I know some of you are also in life sciences, we're also going to talk about image analysis both in cell biology and very briefly in neuroscience as well. And then we're going to talk about using artificial intelligence, and very shortly we're going to talk about the problem of having very large data sets, what to do with them, how to handle those data sets. So Avizo and its sister softwares, Amira and Pore, are essentially Thermo Scientific softwares to visualize, analyze, and understand scientific images. Any kind of image that you have, whether it's 2D or 3D, whatever scale it is, whatever modality it's coming from, whether it's SEM, TEM, optical microscopy, confocal microscopy, tomography images, whether it's micro CT or electron tomography, whether you have hyperspectral images, the software have been designed to handle any kind of images. Avizo is a little bit more geared towards material research. Amira, which is very similar in Avizo, I mean, there are differences, but they're more and less similar to each other. Amira is mostly geared toward life sciences. We also have Pore, which is mostly applications for the field of geology, geosciences, rock sciences, and those kind of researchers. And there's also Open Inventor, which is essentially a toolkit to developing high performance visualization software. We're not going to talk about Pergeus and Open Inventor. We're going to focus on the Avizo. And we're also going to touch a little bit on Amira as well, because these are the softwares that you have available at CEMAS. So anytime that you have an image and then you want to analyze it, it doesn't matter what image you have and what you want to do. This is the process that more or less you want to go through every single time. Sometimes you might skip a step or two, but the process is going to stay the same anytime you have an image. Obviously, you want to start with data acquisition. That goes without saying. And then we're also not going to talk about that. We're going to start at the next step, which is going to be importing the image into the software. For the majority of the cases, it's the data coming out of the microscopes and the CT machines are pretty standard, so Avizo can read them. But even if you don't, if you have a data that is coming from an experimental machine, like a home-built machine or a new format, there should be a way to read it in Avizo. You bring in the data in, and then the next step would be filtering and pre-processing of your data, whether you want to remove of noise, background, or artifacts. The next step is visualization. You want to look at the data that you have. This is especially important in the term of 3D data. You want to look at what you've got just inspected visually. And then we get to a step that is we call segmentation, which is essentially you want to mark the regions that are the different phases. For example, you want to mark cracks or pores or grain boundaries or cells or neurons or a specific kind of a mineral that you see in your images or anything else. This is the process that we're gonna also talk about, is that you're essentially gonna find a way to essentially categorize that image. The most common way is based on the brightness value. The bright areas are one phase, the dark areas are different phase, but there are many different ways to do that. And then once you do that, once you segment the image, measuring and analyzing and essentially quantifying your data is going to be fairly straightforward. And when you go through all of this, obviously you want to create presentation, whether you're generating images for publications or these are animations that you want to create, there are ways to do that. So this is the process that any time that you want to analyze a scientific image, you want to go through it. You want to find a way to import it into the software. You want to filter it, denoise it, get rid of the artifacts. You want to visually inspect the data. You want to segment it, and then you want to quantify it. And obviously, at the end, you want to output what you have done. I'm going to go through these steps in a little bit more details, but the process. This is my main slides. Anytime I talk about image analysis, this is the process that we want to go through anytime you have an image. On top of that, on top of this general process, there are tools in Avizo for fiber analysis, characterization of porous materials, generating meshes out of the images, so converting your images into meshes that you can use for finite element or computational fluid dynamic simulation. You can also use Avizo and Amira for simulation of physical properties. You can do image-based simulation. We're going to go through all of this briefly as well. You can also use the software to essentially track plastic deformation or material deformation and perform what we call digital volume correlation. So if you have an image of before and after deformation, just with that, you can get a 3D strain map of your sample. There are also ways to automate the process, the analysis that you do. The workflows can be automated. And then if you need to, you can expand on top of the software using Python and some other tools that are available. There are ways in the softwares to manage large data. There are ways to automate the process, as I said, and then you can customize it if you want with Python. So the first step is, I'm just going to go through the steps one more time with a little bit more detail. So first is you want to import your data. Any scale, any size, any format, as long as it's an image, you can read it. Whether it's CT, electron microscopy, optical microscopy, it doesn't matter. You can read images. On top of that, you can read surface measures, you can read volumetric measures, and many other formats can be read with Avizo. Even if you have a file that is a brand new format for some reason, you're coming out of an experimental technique, as long as it's an image, as long as there's an X and a Y and maybe a Z and some numbers that indicate a grayscale value or color or something, as long as the file has that format, there should be a way to read that in Avizo. So you're not limited on what kind of data you have and what format it is in. As long as it's an image, there is a way to bring it into Avizo. Obviously, in most cases, you don't even notice it's a step because for the more common imaging modalities, they're either in TIFF or JPEG or some other pretty standard format that Avizo and Amira can just read directly. The next step is going to be visualizing your data. And this is one of the things that is Avizo and Amira are really powerful. Actually, we sell just the visualization engine as a standalone software and that people can use for visualizing other things. So you can visualize your data in 2D or 3D, or a combination of those, or a combination of multiples in 2D or 3D, and then you can make them sit on top of each other, and many other options that you have. You can manipulate it, you can visualize it. There is a lot that you can do to look at your data, to essentially immerse in what you have, to see what you have collected in your data. This is especially valuable when you're working with 3D images. And then obviously you want to denoise your image. There are tools in Avizo for just general noise reduction, the so-called salt and pepper noise. You can get rid of those. There are tools to get rid of the backgrounds that are very common. And then there are specific tools for a specific kind of modalities to reduce artifacts that you see. For example, curtaining that you see, deconvolution if you're working with confocal images, or beam hardening artifact removal if you're working with CT images. So there are all these different tools to make the image as clean as possible without actually losing the valuable information in them. One extra step is that sometimes you want to register your data with each other. Then what's registration? That means that if you have data coming from, let's say, electron microscopy and optical microscopy, or you have backscattering images and EDS images, but they're not exactly aligned to each other. I mean, they kind of are, but they're not sitting right on top of each other. The software can register them together. It can correlate the two images to each other, try its best to essentially make them sit on top of each other so that you can see how does one area look in one modality versus the other one. That is something that you can do. You can also visualize them as we discussed. You can visualize both of them at the same time. And if you need to, you can also stitch and fuse the different volumes that you have with each other inside Amira and Avizo. And then we get to the step that is called segmentation. So we want to go from these images that we have that are all these different phases that we see to an image that essentially makes sense to the software. The most obvious way would be, again, just based on thresholding. You're going to give it an intensity value that it's going to find and say like anything between this value and this value are going to be, for example, in this image of my nanoparticle. Or anything darker than this is going to be my cracks. That would be the most common way of doing that, but that's not the only way that you can do it in Avizo. You can use correlation-based segmentation, use information from multiple images to segment. There is a process we call watershed segmentation that you use not only just intensity, but you also use the edges that you see in the image to essentially guide you through the segmentation. So it would be both intensity and the edges. And then you can use the morphology of the different features that you see also as a guidance to essentially create as accurate of a segmentation as possible. But once we go through this step, once we define the different regions that we have in our image, quantification of the image is going to be, Avizo and Amira are going to be really, really powerful in doing that. there are over 200 native measures that are available, from simple things like, let's say, just like surface area or volume, to something much more complicated, like the orientation or the mean intensity of the grayscale in this region that you segmented, and then everything else in between can also be measured. You can define your very own measures in the off chance that what you want is not included in those 200 native measures. And then you can also perform some statistical analysis on this data that you are generating. So once you go through all of this, you go through all these steps, you obviously want to output. Obviously, you can create snapshots, but also you can create videos out of your images, out of your data. And it's not creating videos is not that complicated. This video and every other video that you see in this presentation, they've all been created just using Avizo, in the majority of the cases by engineers who are just using it for data analysis. So it's not going to be too complicated to generate videos inside Avizo. You can generate presentation scenarios. You can you can control the resolution of the output, both for the images and the videos. And there are many different formats that you can export to. And then if you have immersive devices, you can also export your data to those formats as well. So this is the process that you go through anytime you want to work with the data set. You bring your data, you visualize it, you denoise it, you segment it, you quantify it, and then you add. But on top of this general process that you can do, there are specific applications that might require some other additional resources or tools to help get the best result possible. We're going to go through these steps and then talk about other things that you can do with the software on top of performing your general image process. The first one that I want to talk about is characterization of the porous materials, specifically pore network modeling, which would be a way to generate like a mathematical estimate of the data that you have. You're going to have essentially a model of pores and throats that can be used to essentially get information about the pore connectivity, pore shape, can get information about the flow inside the porous material. Actually, this porous network modeling has been essentially founded in the field of geosciences, for essentially to look at the permeability of rocks. That's where it has been started, but now it has found application in virtually every field of science that works or faces or deals with porous materials. I got one example, which is I thought about giving you an example of geosciences, but it ended up on going with an example from material science. This is an air plug foam that is a porous, obviously a porous foam. On top of everything else that we can do is that once we segment out all those pores, we can use them to generate a pore network model inside Avizo or Amira. We can get information about the pore and throat. All sorts of statistical information can be extracted from this model. You can filter it, just keep some of the pores and not all of them. And then that information can be used to essentially also get an estimate of flow, permeability, and tortuosity out of the data. Another specific application or unique application of Avizo for image processing is simulation. So it's a very specific kind of simulation that Avizo and Amira run. They are essentially all image-based simulations that you can use an image, a binary image, a segmented image that you have to get information about material properties. Specifically, you can get permeability, conductivity, resistivity, and diffusivity. It works directly on a binary image. That's the only input that you need. Obviously, you can control the conditions for the simulation, but the input that you need would be just the image. But this is all an image-based simulation. And then the results can also be visualized in multiple ways. Before I show an example, I'm going to talk about Another way that Avizo and Amira can help you with a simulation, and that is for finite element and CFD simulation, the computational fluid dynamics simulation. Avizo and Amira don't do this. Amira and Avizo are not a CFD or FEA solvers. They're not like ANSYS or NASA. What they can do is that they can generate a mesh from the image that you have. So if you have a 3D CT scan of a bone and then you want to run a finite element analysis on it, what you can do is that you can take that image, you can segment your bone or your particle or anything else, and then you can generate a mesh out of it. Take that mesh out, you can just export it into all the common formats for all the common both FEA and CFD solvers like Nastran, ANSYS, Abacus, and many, many more. You can export in those formats. You can take your mesh, run your simulation outside of Avizo. And then another good thing that you can do is that once the simulation has been done, you can bring the results of simulation back into Avizo because you have this very powerful visualization engine. You can use that to visualize the data that you have. So it can essentially help you with generating the mesh and visualizing the post-processing or post-simulation results. An example of it would be this CT image of essentially compacted silica sands. It can have applications in the field of civil engineering. You can obviously, just like any other process, you want to start with segmentation. So you segment out one region that you want. Once that has been done, you have all these different grains, and then you can generate meshes out of it. You have a lot of control over it about what degree of accuracy and precision you want, how large you want them to be. Also, you don't have to visualize. You don't have to generate a single format for all of them. You can create coarser meshes for one set of for one part of your image, and finer meshes for a different part of your image, because the regions that you are more interested in. So you have all those options. And then I should also mention that this mesh that you can generate, this is going to be fairly fast to generate. It's not going to take days to generate the meshes. You can do all of that, and then you can do the analysis outside of it. Or maybe you don't want to do that. Yeah, obviously you can do that, but Avizo can also run some image-based simulations, the simulations that are essentially based on images, for example, getting permeability. Those are all possible, and there are examples, and then the results of what you have done can also be visualized inside Avizo as well. So these are the results of, for example, flow propagation inside the sample that we have. So all of this can be done with Avizo. You can run some simulation inside of Avizo, or you can generate meshes that you can use to run simulations with other FEM or CFD stuff. Another example of what applications that might need that are probably a little bit more complicated than standard process is going to be the fiber analysis, especially for the times that you have fiber agglomeration, or they are just lumped together, or when you have, for example, CT images, and then the resolution of your CT or micro CT machine is touching on the size of the fibers that you have. So you can't really resolve individual fibers, either because of resolution or agglomeration. This is a very common issue with fiber composite, for example, but that's not the only application that you have. So there is a way to essentially identify and segment each individual fiber, even if you cannot fully resolve that fiber just by looking at it. So there is a way to essentially go around that limitation that you have for this very specific case of fiber. There is a way to handle that. It's a process that we call cylinder correlation. You can essentially extract individual fibers, even from complicated images, like the image that you see here. Once you do that, once you get information out for each individual fiber, you can get statistics, per fiber or per volume or any other format that you want. You can get all sorts of information out of your essentially fiber composite. The example that I have is actually this example is the fibers are more or less visible, but there's a lot more that you can do. So you can essentially extract individual fibers. You can essentially look at their orientation. You can control how they can be colored. You can also, and then you can do much more. For example, you can just keep the fibers that are around an individual the regions of interest in your. Hopefully, I'm not going too fast, but we're going to touch on one of the most interesting applications of the Avizo software, which is something that we call digital volume correlation. If you have an image, if you have a 3D image of your sample before and after deformation, just with those two images, you can track the deformation that is happening. You can track the displacement, and you can generate a 3D strain tensor out of just the two images that you have. This gives you a lot of information about the failure mechanism and the material behavior that you have. This would be also a very good complementary technique to finite element analysis. It's a growing field. It wasn't that common a while ago, but day after day there's more and more publications that are using digital volume correlation to essentially link the microstructure to the mechanical behavior. Avizo has a very excellent way of doing it. It's a two-step process in Avizo, which is the local approach and the global approach. First, let's say you have an image of before and after. You essentially cut your image into a tiny sub-volume, and each one of them is individually going to be correlated. For each one of those, you are going to guess how the volume has before, which is great. It's actually a lot of useful information in it, but it's not complete. It's not the best result that you can get. You can still improve on that, and that's where a global approach is coming in. Is that you use that information, the starting information that you have from your local approach, then you essentially track deformation like in each individual sub-volume. But then using essentially a proprietary finite element simulation, you track the deformation for the whole volume at once. So the whole volume at once is going to be tracked, and then you can see how this image, what you started with, how it has deformed into this new image. Now that you have this deformation tracked, just by correlating those two images, just by guessing how each region has transformed like before and after a deformation, just by images. Now that you have that, you can get all sorts of information about both displacement and strain out of it. And you can get it not for the full volume. If you have segmented your image, you can get information out for very specific regions. For example, you can just track deformation, for example, just around the tags that you have. Or if you have a lot of voids in your sample, you can look at those regions. You can look both at displacement as well as your essentially volumetric, the strain before and after deformation. And that tells you a lot about how the deformation is happening. So these are the things that you can do. And then I have a few slides about what you can do with the Amira software, which you also, I believe, have access to at CEMAS for cell biology and neuroscience. And then you probably know Amira software is not a diagnosis software. It's not for diagnosis procedures. This is just a research-only tool, but it's great for doing research. For cell biology, you can essentially get all the benefits of Amira for processing and segmentation of the images. You also get access to a tracking solution for tracking objects, or specifically cells. You can track cells. It also is great for registration method to essentially to get very good registration with Amira for cell biology. Also with Amira for cell biology that you have, you can essentially read all the very common file formats that are common in the life sciences, anything from DICOM to DTI images and MRI images and many more kinds of data. One of the great things about Amira for cell biology is going to be object tracking, which is going to be essentially you can track the movement if you have a time series data. So if you have a 3D data set that also is a time series, it's technically a 4D data set. You have X, Y, Z, and T for time. You can track the different objects inside of it. It's a fully automated tracking solution. It precisely tracks thousands of cells, and it's you can select the motion models. You can have event detection like disappearing or appearing if they are merging together, or if a cell is separating. And once you do that, and once you create the track, there are over 40 track measures that are available for quantification. For example, the speed, the shape of the track, and many more. Amira also has tools to essentially work for some of the very common file formats that are unique to the field of biomedical brain research, mainly the DTI or diffusional tensor imaging fiber tracking workflow that essentially you can input the gradient images and it gives you the actual results of the DTI, including the tensors that the software can calculate all of those. And then again, as we talked about everything else, that it's flexible in terms of visualization. You can both visualize the results as well as the tensors that you have. You can both visualize the fiber tracks as well as the tensor fields that you have generated during the DTI calculation. One last topic that I want to talk about is going to be about the challenging data that you have. Sometimes all of these tools that we talked about are great, they're fantastic, but then sometimes you just lost about how to use them. A very good example would be the shine through or Pore bank. You have an image like this that is a porous, solid oxide fuel cell. It's an SOFC. You have all these pores in it. Some of them are easy to segment. So this is going to be really easy to segment. But what about this pore? I look at it, I know it's a pore, but the grayscale value inside of it is the same because the layers below it are going to be visible. So here, it should actually be dark, but because we can see the layers behind it, it appears to look like this. This is going to be such a difficult job to segment out this region. To differentiate between this is using the conventional ways of doing segmentation. It's going to be such a big headache. That is where deep learning and artificial intelligence is going to come in. Like so far from the beginning, when everything began, people went like in the 80s and 90s, when people were thinking about artificial intelligence, they were thinking, okay, we have a problem, we're going to define a solution for it. If we define a good enough solution, it can find an answer for us. That's how everything is done. That's how most of the stuff that we were talking about is working. You have an image, you know what you want to do with it, you tell the software how to do it, and then it gives you the results. With deep learning, you're actually gonna do it in a different way. You're gonna have the original image, you're gonna give it the answer, you're gonna give it the right answer, just manually go tell it where all the pores is, and let the software figure out the solution by itself. So instead of giving it the solution and asking for the answer, you're giving it the answer, and the software will give you the solution. But once you have that solution, you can use it on any other similar kind of image that you have for simulation. It's much more complicated than that, but the basic idea is exactly like that. You have an image, You give it the you give it the answer, it figures out the solution. This is how it's going to work for deep learning. I have this image, and then I have my not so good segmentation of the pore. I'm just going to go ahead and then label out with whatever technique that you want. This is fully manual segmentation, just painting over them, just cleaning it. Or you can use any other way that you want. You can use this labeler to essentially define all the regions that are going to be your essentially actual pore. So I'm just giving it the answer with the most basic way that I can. But then once I have the answer to my problem, finding the solution can be done using deep learning. So that input painted that we call that solution, or we can call it the ground truth, that is going to go through a process. It is going to be fed into the software. The process is actually more complicated because you also have verification and validate. You have to verify and validate your results as well. But essentially, you have this input data. You have your ground truth. It goes into the software. It predicts. It tries to minimize the error and essentially optimize the accuracy. It's just going to go through an iterative process. It's just going to repeat it and repeat it and repeat it, and it's going to give you a model. It's not going to be a model that is comprehensible to humans, but it's essentially like a multi-layer neural network with hundreds, if not thousands or hundreds of thousands of parameters. But once you have that model, any other time that I have an image that looks like this, I don't have to manually segment that anymore. I just put in this model, and instead of getting this half accurate result, I'm going to get the full accurate result. In this very specific example, the porosity was around was off by like 7%, which is a lot. But I mean, like, you can fix it. And then, so if you have, and by the way, this was a 3D data. I have done it on a few slides, and then that has been used for both validating and verifying the results. But then once this model is available, every other time that I have this image, an image that looks like this, the solution has been generated by the software. You don't have to do it every other time. So this is also available in Avizo and Amira for any kind of for the super challenging images that you have. I'm going to finish by talking about handling large data, and then just a very quick slides about the other things that are available. So large data, what is a large data? Anytime your data is larger than the memory of your computer, it's a large data. If you have this CT of a complete car, it's probably a few terabytes. That is a large data for any computer. If your laptop has 4 gigabytes of RAM, a data that is 5 gigabytes is a large data. There are ways in Avizo and Amira to essentially both visualize and process the data, even if they are larger than your RAM on your computer. Even if you can load the data fully, there are ways to do that. There is an out-of-core technology that is available that you can process the image like one slice at a time, just with one slice, process it, output it, without actually loading the full data. That's one solution. There's also an LD format. LD stands for large data. So there's a large data format that is also available that is essentially going to convert your images so that the file is readable. It's a little bit different, but I mean there's a way to read it into the software. If you do that, you can visualize that data easily, and then you can do not all but many of the processing on this very large volumes that you have that are sitting over sometimes even above 300 gigabytes. That number needs to be updated, but I'm pretty sure we're getting to like maybe 2 or 3 times larger than that. And then the very last slides I'm just going to go through very fast. You can automate the whole process. There are many ways to do that. One way is to call this creating recipes. You do the analysis on one sample. The software is going to remember all that you did. And then the next time you have a data set, you can load it. Obviously, you can fine tune it if you want to, but the process is going to be that you can repeat it on any other similar data that you have. If you want to customize and expand on the software, this is obviously not an open source software, it's a commercial software, but it's not a black box. You can use Python to expand on it. You can use C++ to expand on it. And then if you're working with MATLAB, you can force Avizo and MATLAB, or Amira and MATLAB, you can force them to talk to each other, because they can essentially build a bridge between MATLAB and Avizo, for example, for transferring data, or do some of the stuff in MATLAB if you want to. Those are all possible in Avizo, and it would be well integrated just like any other step that you have in your process. And finally, the software comes with a lot of support. So once you get the software, that's at the end of it. There are product experts that are going to help you with the process. There's a hotline that you have access to. If you have a question, you can email them and ask them, hey, what is that I heard about the cylinder correlation? I run it and I get bad results. Do you have any suggestions? So you have access to that hotline. It's not actually a hotline, it's an email. It should be called actually a hot email. There are trainings like the ones that we did last year and also this year virtually at CEMAS, both introductory on-site and here in Houston. There's also consulting and custom development available. And there are online resources. There are videos on YouTube that are quick, on point, not too long, 5, 10, if 12, 13 minutes, if that long. They're about a single topic, and they are just on point. They are not marketing or anything like that. Those are instructional videos that you can use. We have a use case gallery that is essentially a collection of a constantly evolving collection of not all the papers, many of the publications that are using our software. And recently we have our extra library. It's extra, so it's xtras.amira-avizo.com. It's a collection of custom script, deep learning models, guides, a very detailed script description, and many more stuff to do. So it's like a collection of everything that you can do with the software, like examples for segmenting the image, generating meshes for digital volume correlation, example of how to get the volume from a fiber, and many other things. This extra library that we have recently launched, this is a very great resource, and it's evolving, and it's evolving fast, and there's always something there that it would be useful for you. And with that, I'm going to finish my talk. Thank you for listening, and then we can jump to Q&A.
Thanks, Ali, for the talk. I think I learned a bit about some more capabilities I didn't know about Avizo before, so that was great to hear. If anyone has questions to start with, go ahead and type those in the Q&A, and we'll start answering some of those.